what I'm going to tell you about is the design of proteins that carry out new and useful functions. So essentially all the important processes of life are mediated by protein molecules, and these protein molecules evolved in response to all the challenges that were faced during the evolution of humans and the evolution of all organisms. And the more you understand about biology, the more you realize how amazingly well the proteins that exist in nature solve these problems. But today we face new problems, and we're heating up the planet. There's new viruses, new diseases. We're running out of fuel, and again, appreciating how wonderfully the proteins that exist in nature solve the problems that arose during evolution, one can have confidence that in about several billion years, we would have new proteins that would solve modern-day problems as well as our naturally occurring proteins solved the problems that were faced during evolution. But of course, most of us don't want to wait 3 billion years for problems to get solved. And so that's the idea of protein design. We're trying to develop methods for designing new proteins that solve current-day problems, as well as the proteins in our bodies and the bodies of other living things solve the problems that arose during evolution. So first, I need to tell you what proteins are before I tell you how we designed them. So as I said, proteins mediate the critical processes of life. Proteins are made out of amino acids. There's 20 different types of amino acids, and each protein has a unique sequence of amino acids. So a protein typically would have about 100 amino acids, and they'd be strung together in a linear sequence. So it'd be like one long 200 or 100 letter word. So each protein has a unique structure that's essential for carrying out its function. And here I just show pictures of different protein structures that carry out different functions in our bodies. I told you that each protein has a unique amino acid sequence, and so you can represent these sequences kind of like beads on a string like this. So they have names. The names of the different amino acids are indicated here. Probably if you go to a health food store, they'll try and convince you to buy a jar of one or a variety of these, but it won't do you any good because you eat proteins every day anyway. So each protein has a unique sequence of amino acids. Actually, in this picture, the same amino acid has the same color. So you can imagine 20 different colors or 20 different. They're 20 different types, and they're just strung together in a linear sequence like this. So you can imagine how is it hard for how does a particular sequence like this do anything useful? And it's because, as I said, it folds up to a three-dimensional structure like this. And the interesting thing is that each different amino acid sequence folds up to a different three-dimensional structure. It's not obvious, however, how the sequence of amino acids determines the three-dimensional structure. So that problem of predicting how the amino acid sequence of a protein determines what its three-dimensional structure is called the protein structure prediction problem, and that's a problem that I worked on and my group worked on when I first came to the University of Washington. So, but there's another way of looking at this problem, and we can, and I guess maybe I'll just go to the next slide to illustrate it here. So, where do amino acid sequences come from? Well, you all know that we have genomes. We're in a genome sciences department, and in each of our cells we have a genome. A genome is made out of genes. There are about 30,000 genes in our bodies. And each genes are made out of DNA. You've heard of DNA. And each gene encodes a particular sequence of amino acids. That sequence of amino acids, as I just told you, will then fold up into a three-dimensional structure like this one. And so this is essentially all of biology is on this slide. We have DNA, genes, encoding amino acid sequences. The amino acid sequences then fold up to proteins, and the proteins then go and do mediate all the important processes of life, like I said. As you're hopefully listening to me, there are ions passing through proteins in your brain that are producing currents that are giving rise to your thoughts. If you ate before, there's proteins in your stomach that are breaking down the proteins in the food that you ate. And if you walked to get here, there was proteins in your muscles that got you here. Okay, so this is basically biology. We have genes going to amino acid sequences, going to protein structures and functions. But now let's think about, and this is called the central dogma of molecular biology. The information goes this way. But now, if we understood how to go from DNA to amino acid sequence to proteins, you could imagine running this process in reverse. And I'll illustrate that now with this protein here. This protein turns out to not be a very nice protein. It's the protein that's on the surface of the flu virus, of the influenza virus. And so there's flu strains, different flus every year. This actually happens to be the protein that's on the surface of the 1918 pandemic influenza virus. But it turns out that all influenza viruses have a very similar protein on their surface. Okay, so this is not a good protein. But what if we understood how to go backwards? Then you could imagine designing a brand new protein that doesn't exist in nature, this little red thing here, and you could design it, if you knew what you were doing, in such a way that it would stick very, very tightly to this flu protein, and if it stuck tightly enough, it might prevent the flu virus from getting into our cells. So you start with a computer model of this protein bound to, the design process is, the design challenge is to design a protein that will block the flu virus from getting into cells. So once you've designed the protein on the computer, what do you do then? Well, now you go backwards. So you've designed this protein on the computer, and I'll tell you in a moment outline how we do that. And now you have to go backwards to the amino acid sequence of the protein. So we designed this protein so we can, in the process of designing it, what we did is we found an amino acid sequence which predicted to fold up to a structure that is predicted to block the flu virus. So in fact, after we build, maybe I'll just say that again, after we come up with a computer model of a protein that would block the flu, I'll actually show you an animation in just a moment that shows how we compute a sequence which is predicted to fold up this way. Now, then we have an amino acid sequence. To go back to the DNA, that's called the genetic code, and that was worked out in the 60s. So once we have an amino acid sequence or once we have a protein structure on the computer, we can work out what amino acid sequence it would have. And then we can, by using the genetic code, identify a DNA sequence which encodes this protein. Now, this is a brand new protein, so that DNA sequence doesn't exist in our genomes, but it turns out that DNA is essentially a commodity item. There are many, many companies today who compete with each other to offer you more DNA at a lower price. And so every month we look at all the different companies and see who's giving the best deal on DNA. And then we order lots and lots of synthetic genes, pieces of DNA that encode these made-up proteins we've designed. So after we have the amino acid sequence, essentially, I just did this the other day with proteins I'm designing. We take that amino acid sequence, we put it into an email, and we send it to the company that's giving the best prices that day. And we can now do this for, it costs about 50 cents per amino acid, so it's getting cheaper and cheaper. So now, once we have the DNA, this artificial gene, it's not a gene in our genomes, but a gene that encodes this protein, we put the gene into bacteria, and the bacteria make lots and lots of the protein. And then we can get after we've grown up the bacteria, we can break them open, and the protein pops out, and then we can see whether it in fact blocks the flu virus. And that's basically what we do for every problem I'm going to tell you about today. Now, why is, you might think, well, there's so many organisms in nature, and we have 30,000 proteins. There are, there's like millions of different organisms. What's the point of designing new sequences? Well, it's useful to consider the number of different amino proteins that could exist. So I told you there are 20 different amino acids. So at the first position, you could have any one of the 20. Then at the second position, you could have any one of the 20. And at the third position, you could have any one of the 20. So the number of different protein sequences is 20 times 20 times 20 on to the number of amino acids in the protein. And proteins, as I told you, typically have 100 to 200 amino acids. So the number of amino acid sequences, which is the number of different proteins which are possible, is 20 times 20 times 20, 200 times, which Ella can tell you is a very big number. Nature has only explored a tiny, tiny fraction of that. So maybe nature has explored 10 to the 10th sequences at most, and the number of possible sequences is something like 10 to the 200th. So nature really hasn't gotten anywhere in exploring what's out there. And that's what we get to do. We get to try and find things that don't exist and then make them and see what they do. Okay, so how do we actually do this? So this is what we do when we're designing proteins. We start with a computer calculation of a sequence which is predicted to fold up to make a protein with a structure that we want or a function that we want, like blocking the flu virus. As I said, once we've designed it, we know what the amino acid sequence is. We can use the genetic code to get the DNA sequence and make the gene, and then we make the protein and assay it. Okay, so now I'm going to explain to you a little bit about how we design proteins. And the concept I have to explain is that the way that an amino acid sequence determines a three-dimensional structure is that the amino acids, the structure that the protein folds to is the lowest energy structure for that amino acid. So if this were a bumpy surface and there was a ball rolling around on it, then this would be the lowest elevation point. But this is instead a protein. It's not rolling downhill literally, but it's going downhill in energy. And what that means for those of you who know a little bit more about interactions between atoms is that the atoms in the protein are getting close to each other. There are hydrogen bonds getting made. Opposite charges are getting close to each other, and that's why the energy drops. So to design a protein, if we want it to fold up to this structure, we have to find a sequence such that this is the lowest energy structure, that this structure is the lowest energy. Okay, so now I'm going to show you in sort of an animation of a computer calculation where we say we want to make a protein that has this shape, and this is just showing where the backbone of the protein is. And these little squiggly things that are coming in and out, the little stick-like things, are different amino acids. So amino acids differ by having different what are called side chains coming out. And so when we're changing the sequence, we're just changing the shape of the amino acids, the identity of the amino acid that comes off the backbone. And when we're doing this calculation, basically what the computer is doing is searching for the combination of amino acids which give this structure the lowest energy. Okay, and so, but one thing you can see here is that we kept, the protein was kept fixed. We didn't really change its overall shape at all. We just changed what the amino acids are. So this will give us an amino acid sequence which is very low in energy in the structure, but there might be some other completely different structure which is lower in energy for that same sequence. So once we have that sequence, we do a second calculation where we keep that sequence fixed, but now we search through a very large number of possible conformations for this sequence, different shapes of the protein, looking for what the lowest energy shape is for that sequence. And what we are looking for is sequences that have the property that when you search through this very, very large space of possible conformations, the lowest energy one is the one that we want. And that's basically how we do protein design. So now, the rest of my talk, I'm going to give you examples. So the very simplest problem you might think of is sort of what I was outlined in those two movies. Can we simply make a sequence which will fold up to a new structure that's not found in nature? Now, proteins in nature, because they evolved, are essentially all accidental. They have, each one has kind of idiosyncratic features because nature never really cared about elegance. Nature just wanted to get something that worked. So we decided to try to make sort of minimalist, idealized proteins, and it's a little hard to explain that if you don't know a lot about protein structure, but let's just say they're represented by these schematics here. Now, we then run that first movie to find an amino acid sequence which is very low in energy in this structure or in this structure. And now we want to check that amino acid sequence has no other structure which is still lower in energy. Now, it turns out this is a very big computer calculation because a protein chain is very flexible. There are many different possible conformations it could have. So a number of years ago, we started a project called Rosetta at Home, where we enlisted volunteers all around the world, and we now have 350,000 volunteers. And if you get excited about what I'm talking about, maybe we'll have 350,050 by tomorrow. Anyway, you just go to the website, you sign up your computer, and then what happens is, what happened in these cases, for example, we take the sequence of this protein and we send it out to all the volunteers in the network, all these 250,000 volunteers. And what they do is they search. They run that second movie. They search. The protein folds up on their computer, and they send us back the lowest energy structure that they find. So this is the energy, and this is the distance from the structure we were trying to make. So some people are unlucky, and the lowest energy structure they find, I should say each red dot is a different person's contribution. So you can see we have a lot of volunteers. So some people end up pretty high in energy. This is energy on this axis. Remember, the search is trying to look for the lowest energy state. And when they're high in energy, they're often far away from what we were trying to make. But what you can see here is the people who got really low energy structures also got quite close to the structure we were trying to make. So this sequence has the property that when you use 350,000 people's computers all around the world and try and predict what its structure is, the lowest energy structure they find is really close to what we were trying to make. And so then, if we have a sequence that has that property, we then, as I said, we order a gene that encodes that protein, and then we make the protein in bacteria. And what we find when we do this is the proteins that we make are very, very stable. It's very hard to pull them apart, much harder than it is to pull apart naturally occurring proteins, because again, naturally occurring proteins didn't evolve to be optimal in any sense like that. And when we use experimental methods to determine the three-dimensional structure, we find that the proteins that we've designed are very, very similar to the design models. So we can design brand new proteins that have exactly the structure that we want them to. On the other hand, this is kind of boring because these proteins don't do anything. They're very, very stable. So it's like we've designed new rocks, with very particular shapes. And we've gotten very good at designing rocks with, well, very small rocks made out of protein with very different shapes. And the project I'm working on is making very long, skinny ones. And I won't tell you why I'm doing that. Okay, but now you're going to be more interested in proteins that can do stuff. I mentioned earlier the flu virus, and this is a picture of what influenza virus looks like. It's got these things on its surface are proteins that target the virus to receptors in our lungs and then help it get into cells. And this right here is a blow-up of what this protein looks like. And I showed you another representation of that same protein earlier. Now the flu virus is changing all the time, and that's why we need a new vaccine. But there are two regions where it just doesn't change, and that's these yellow regions here, one here and one here. And so, as you can imagine, when we looked at this problem, we thought, well, how about we try and design very small proteins that block either this site or this site? And I showed you already, and here's another cartoon of a protein which we've designed to bind to this site, which doesn't change in the flu virus. And this is now this yellow here is really a blow up of the surface of the virus, and this magenta thing is one design protein, and this is another design protein, and these things coming off here are the amino acids in the design protein. And what you can see, even if you don't know anything about biology, is that in the shapes of the amino acids coming off this design protein really fit very nicely into grooves in the surface of the influenza virus, like this. And all of molecular recognition in biology, almost all, is mediated by really very simple complementarity in shape between to proteins. Usually, in the case of what the influenza virus is trying to do, it's trying to get into your cells. So it has its receptor is very complementary to something that's on the surface of your cells, and they just get very close and they fit, so it sticks. Here we're using the same principle of molecular recognition, but now to fight the flu virus, to make things that are very complementary in shape to its surface. Basically another way of saying that is that this is a very low energy arrangement when this designed protein is bound to the virus, and so it goes there and it stays there. We made these proteins, we made the genes encoding these proteins, we made them in the laboratory, and we found these proteins bind very, very tightly to the virus. And this is kind of a complicated slide, but this is another picture of that viral protein. And in purple here is the design model, our computer model of where this designed protein was supposed to bind the viral protein. And in red is the experimentally determined. There are experimental methods for determining protein structures, which are very complicated and difficult. But those were done with these design proteins. And what you can see is that where this protein actually binds the virus, which is shown in red, is really almost identical to where it was designed to. And that even comes down to where the shapes of these side chains are. So this computer model we made of a protein binding to the virus came out almost exactly right. Now, to actually see whether the protein helps prevent the flu, that's not something we can do in my laboratory, but fortunately at the University of Washington and in Seattle generally, there are many, many people who study the flu and other things. And so, and Deb Fuller's lab in the microbiology department in particular does, and her graduate student Marika took this flu-binding protein and showed that whereas when mice got treated with the flu, they lost weight and nothing good happened to them after this. When they got this flu-binding protein at the same time, they were just totally normal. And it's kind of neat the way that she's giving them through the usually drugs are administered by protein drugs are administered by giving an injection. Turns out these proteins, since they're small, can be delivered intranasally, and we're hoping ultimately by inhalation. So maybe in a few years you start feeling sick, you can go to the drugstore and inhale a little bit of one of these proteins, and it will wipe out your flu virus. But we're not quite there yet. Okay, so this basic idea of designing proteins to bind to other proteins, I described this case of the flu. There are many other types of viruses and bacteria that we're applying the same approach to try and make sort of a new class of drugs that will prevent infectious disease. There also are other diseases like autoimmune disease and cancer where your cells go out of control. And so we're designing proteins which will bind to the surface of tumor cells, for example, and shut them down. But since I've already given you one example of proteins that bind to proteins, I'm not going to talk about that. I'm now going to talk about something a little bit different, which is proteins that bind to small molecules. So this is a small molecule which is called digoxygenin, which is used to treat certain types of heart conditions, heart disease. It has the problem, though, if you get too much of it, it can kill you. And the range between where it helps you and where it kills you is not very big. So we want to know is whether we could what we tried to do is design a protein which would bind to this and soak it up, so that if you were a doctor and you gave a patient too much of a drug like this, you could then remedy it by giving them the protein, giving the patient the protein. So the process we used is very similar. Now here's this thing we're trying to bind. It's not a protein, it's this small molecule. And the same kind of thing, we designed basically interactions that the design protein here is in green. We design amino acid sequences that interact, make very low energy interactions with the small molecule. And here's a small molecule, and here's our designed protein, and it really fits very snugly. It's kind of like a hand in a glove. So again, this is just a computer model. We then make the gene that encodes this protein. We make the protein in the lab and see whether it binds the small molecule. And the protein does bind the small molecule. It actually binds the small molecule very, very tightly. And here's again when the structure was determined using experimental methods. That's what you see in cyan here. And the computer model, the design model, what we were trying to do was in magenta. And you can see the designed protein is coming out right, and this small molecule is bound in pretty much the right way as well. So there are many small molecules where it would be very useful to have high affinity binding reagents. Some of them are other drugs like apixaban, which is a blood thinning reagent, which also can be lethal if you get too much. There are other compounds where you'd really like to be able to detect them, to know that they're there. So things like hormones that are associated with stressful conditions, or one of the compounds we're trying to make where we have binders to now is called artemisinin, which is a very promising anti-malaria drug. But it turns out there's a big problem with counterfeit artemisinin in the world today, because people sell, if you're buying it, you don't know what you're getting. So we've got a protein now that binds quite tightly to artemisinin. We think that could be used to validate whether what you're actually getting is the right thing. And sort of on a similar vein, Washington State has asked us to design a protein which binds to THC, which is the active ingredient in marijuana for obvious reasons. So anyway, so there's a lot of different, both for sensors and for what I sort of generally call therapeutic sponges, binding up toxic compounds. Also pesticides. We've designed proteins which bind to toxic compounds and can soak them up and detect if they're present. But I won't talk any more about that since I already gave you an example, this one example. The next thing I want to describe to you is the design of materials. Now you might, so why are we interested in materials? Well, some common things that you're used to, like silk and wool, are made out of proteins. Other things that you're familiar with, for example, the shells of oysters and abalone clams, are made out of, and essentially all materials all materials that occur in nature are not made out of protein per se, but they're organized by proteins. So if we could figure out how to make, how to take the types of, you can think about all the sort of exotic materials you have in nature, ranging from shells to silk to elephant tusks. If you could figure out how to make those things synthetically, there you could imagine a whole new generation of types of materials and products we could have around us. So I'm going to tell you about how we're going about that. So in a material, it's not just one protein typically, it's many proteins interacting together. And so first I'm going to describe a material that's made out of, that's rather than a continuous thing, it's a closed cage. Here we're taking two proteins, a green protein and a blue protein, and figuring out how to fit them together so they make this very, this closed structure here. And again, we can find the way we make these proteins stick together is we design a set of we design the amino acids at the interface between the blue and the green protein so they stick together. I'm not sure how well you can see this, but when we make these two proteins in bacteria and we break the bacteria open, when we look in an electron microscope, what we see are, this is the computer model of what we were trying to make. And what we see are these things that look like little shredded wheats. They look this one's actually a pinwheel more. And we get these particles which look almost identical to what we were trying to make. And it's a little hard to see, but that's true for each of these cases. So we can design proteins that assemble together to make closed shapes. And we can go further using an experimental method called X-ray crystallography. We can actually determine where every atom is in these designed nanomaterials. And it turns out that the atoms are nearly identical place in the actual structures as they were in the computer model. So we can make computer models of these new types of structures, and then we can come up with amino acid sequences which fold up to these very precisely. And here's the pinwheel one, the design model, and here's the crystal structure. And so we're making more fanciful ones now. This is one that's an icosahedron. It basically looks like kids' jungle gym, the ones that have all the bars that go together at the top, except this is a complete icosahedron, and this one also assembles into icosahedral particles when you make it. Okay, so what are we trying to do with these? This shows pictures of some of the materials we've made so far. You could imagine a number of things, and these are the kinds of things we're working on now. These are like containers. So to treat conditions like cancer, currently, as you know, people are treated with chemotherapy agents. They're drugs which are very, very toxic, and you take them and they go all over your body and they kill the tumor, but they also do a lot of damage elsewhere. So we're working on ways to package inside these particles the chemotherapy agent and then put on the outside proteins which bind to the outside of tumor cells, so the chemotherapy agent will only go where it's needed to go. Another exciting possibility is these. I didn't show you what a picture. Well, I showed you that picture of the influenza virus, but I didn't show it very precisely. What a virus looks like is sort of like one of these big cages with things sticking out. So we can make things that look like viruses, and in fact have viral proteins sticking out, and maybe make a whole new type of vaccines. Currently, vaccines are just basically killed virus from last year. But as we learn more about the immune system, we should be able to design particles that elicit an even stronger immune response to the virus. And now this is now something which is not a closed structure, but an open structure, a layer. And for layers, now we're starting to think about templating things like abalone shell, where you could imagine a layer that then leads minerals to be deposited down upon it, as well as nanowires for nanoelectronics and other related topics. I'll show you a picture of a layer in a minute. So this is a protein which has been designed to interact with other copies of itself to form regular lattice. And when we make the gene for this and break open the bacteria, we see that the protein does indeed form a regular lattice that is very similar to the design. So now we're trying to think about how to detach things to it, so we can start making this layer do things. Okay, so I'm just going to talk for about 2 more minutes. So I mentioned already Rosetta at Home, where all the calculations I've described are very computer time intensive. And so while we've tried to put computers in every available spot in the University of Washington, we kind of ran out. So that's why we needed to list these 350,000 volunteers. But once we had it going, if you participate in Rosetta at Home, there are message boards, and people started writing in, and I have sort of a blog there. People started writing in and saying, well, I'm watching my computer fold up the protein. You see, when you run Rosetta at Home on your computer, you see a screensaver that's basically like the movies I was showing you. It shows you what the computer's doing. And they would say, well, the computer keeps moving this part of the protein to the left, but it really should go to the right. And can't you make it so that I can help guide the computer so it can do a better job? And so this led to the idea of making sort of an interactive version of Rosetta at Home. So we teamed up with the people in the computer science department here and developed an interactive game called Fold It. Now in Fold It, it's very much like Rosetta at Home. We're either we're trying to design new proteins, but now the player can help guide the computer. And so this is what Fold It looks like. And again, if you just Google Fold It, you can install it on your computer, and it gives you a set of tools which are basically the operations I've already described, like let the protein fold up. But you can also go in and you can pull and push things around and fold up the protein on your own. And so good Fold It players know how to go back and forth between sort of moving things by using their intuition and then using these various tools. So this was actually an interesting one. This was a protein which no one knew the structure of, but was a monkey virus protein. So we called this an unsolved monkey virus protein puzzle. And it turned out the Fold It players actually managed to determine what the structure of this was, which was very exciting. And they've actually solved a number of different problems, including designing. I didn't talk about one of the things we work on is designing catalysts for chemical reactions which don't currently have catalyst. A catalyst is something that makes a chemical reaction go faster. And folded players have improved some of the designs we've made. But rather than tell you about that, I thought people always ask me, well, who are these folded players? And I thought I'd let them speak for themselves.
A really good folded player, I think, needs a number of skills. Problem solving is obviously one of them. I think a slightly addictive personality, a little bit of stubbornness thrown in, and a little bit of luck on top. You don't necessarily need to have a scientific background to play. You just need to know what works and what doesn't. And you can learn that within the game itself.
I'm an administrative worker in the rehab team. I'm just answering telephones, working on bespoke computer programs, interacting with staff. It's very repetitive. When I go home, I become a different person. I just like to measure myself against something, and it's given me something that my everyday life hasn't given me. It's to just use abilities I didn't know I had.
For me, it's a guilty pleasure, and yet here I am involved in something that has real relevance in the scientific world. It makes you feel very proud of what you do, which is essentially a little hobby.
He's much more professorial than I think I will ever be. So these were actually two of the top folded players in London, which is where the video was made. And so they asked us where, for the names of folded players in London where they were making it. Okay, so I'm pretty much done. We've started an Institute for Protein Design at the University of Washington to try and really fulfill this vision of designing proteins to address modern-day problems. And these are the different areas that we're working on, trying to make improved therapeutics, both improved drugs to treat infectious disease, as well as conditions like cancer. I mentioned diagnostics, detecting toxic compounds in the environment, improved vaccines. We're working on new ways of fixing carbon dioxide and making fuels. I talked a little bit about targeted drug delivery, and I mentioned new materials. These are all areas that we're working on. And somewhere here, I think I have some more information on it, or you can look it up on the web if you're interested. Now, I didn't really do any of this myself. I'm very lucky to be working with some really fantastic co-workers, graduate students and postdoctoral fellows. And I won't go through everyone's name on this slide, but these are the people who did the work on the design of the different proteins I described. And again, I'm sorry about starting late, and I'll be around for a bit if you want to, or happy to take questions now first and talk to people in person afterwards.